Hi there. Welcome to Labster. In this lab, you will learn about different bacteria shapes and their different methods of moving by studying a bacteria sample using a microscope. This simulation uses voiceovers. If you don't have audio, click the speaker symbol on the top right of your lab pad to switch the play mode. If you disable the sound effects, the lab pad will slide up whenever there are instructions to read. You can select what type of audio you prefer. Deselecting any of the options will mute that option. Click Continue to learn how to navigate through your mission. Using the lab pad is an important way to get the most out of this simulation. Is this the first time you've played a Labster simulation? Perfect, then let's get started with your first mission. Our friend Nicholas is a researcher in Antarctica. He has collected a sample of ice that contains bacteria. Today we are going to study the shapes and movement of the bacteria with a microscope. While I prepare the Antarctica sample for you to study, let's get you sorted with a lab coat and get some gloves on. Pick up a lab coat from the rack. Slide up the lab pad for more info. Well done. Now find a glove box and click on it. Slide up the lab pad for more info. Great. Now we can get started and study the sample in detail. Go to the microscopy workbench where I have placed the sample from Nicholas. Go to the microscopy workbench. In order to study the bacteria in more detail, I prepared a more concentrated sample from the one Nicholas gave you. I centrifuged the sample, then, I removed the supernatant and added a smaller volume of water. I have placed the 15 milliliter tube on the table. We will use a single cavity microscope slide for viewing the bacteria under the microscope. This is because then we don't need to fix the bacterial sample, and can instead view the living bacteria moving around. Can you prepare a single cavity microscope slide with the sample for us to look at under the microscope? Pick up a single cavity microscope slide from the stack. Put the slide on the tray before you add the sample. Use the pipette to add the sample to the slide. Discard the tip in the trash bin and place the pipette back. Place a cover slip on the microscope slide. Place the slide in the slide holder on the microscope. The microscope is a light microscope which can be set in bright field mode or dark field mode. The microscope is connected to the screen, so you will be able to see the different modes by clicking on the buttons on the screen. Keep in mind that normally you would not see the flagella structures of bacteria under the light microscope. However, for the purpose of this experiment, we have made these structures visible. To see the flagella in the real lab, you would have to use the confocal microscope. Can you decide which setting is best if we want to look at the bacterial shapes and movements in our sample? Check out the two settings on the microscope screen and raise your lab pad when you are finished exploring. Are you done exploring? Great. Then let's carry on with this experiment. Which setting is best to view the moving bacteria and bacterial shapes? Awesome, that's right. Let's go with the dark field mode. Sometimes it is easier to see in bright field, but it depends on the sample and what you are looking for. Great work. Now we can see what's in the sample we got from Antarctica. Let's see what types of movement we can detect. If you click on the microscope, we can jump into the sample solution. Click on the microscope to get a closer look at the bacteria sample. Find out more about how certain bacteria move by clicking on any in the sample. This bacterium looks like it's drifting, but it actually has internal flagella embedded into the cell wall. These structures are called axial filaments that are unique to this type of bacteria and generate a corkscrew-like motion. Find out more about how certain bacteria move by clicking on any in the sample. This type of bacteria moves via short appendages called pili. They look similar to fimbriae and cause the bacterium to move in short intermittent jerky motions. They are also important for attachment to surfaces. Find out more about how certain bacteria move by clicking on any in the sample. This type of bacteria moves around with a tail-like structure called a flagellum. The structure of the flagellum itself is quite advanced and consists of multiple subunits which cross the cell wall and are anchored in the plasma membrane. Find out more about how certain bacteria move by clicking on any in the sample. This type of bacteria have a small cluster of flagella, which rotate in a coordinated fashion like a propeller. When the bacterium is running in a certain direction, its flagella are organized into a coordinated, corkscrew-shaped bundle. However, when the cluster of flagella rotates in a clockwise direction, the flagella separate as independent units, which causes the bacteria to tumble, instead of moving in a single direction. The tumble results in a random reorientation of the bacterium so that it often ends up facing in a different direction. Therefore, when it next begins to move, it usually goes in a different direction. Rock on. You are becoming a skilled microbiologist. The bacteria in our sample have different movement patterns. Why do bacteria move? That's right. Bacteria can be attracted to different nutrients or environmental cues such as temperature, light, oxygen, osmotic pressure, and gravity. However, they can also be repelled by toxic environments. They can also move towards each other and form biofilms that provide them with additional characteristics. Now we know that some of the bacteria we found can actually move around to form colonies with their peers, to get to the necessary nutrients and avoid toxic environments. Pretty smart little guys. The bacteria that we have from Antarctica also have different shapes. Can you help me catch one of each and annotate their shape correctly? Click on any of the bacteria and identify its shape. You can always look at the theory or image tabs if needed. Which shape is the bacteria you caught? Yes, absolutely. Rock on. This is a Vibrio bacterium because it's comma-shaped. Click on any of the bacteria and identify its shape. You can always look at the theory or image tabs if needed. Which shape is the bacteria you caught? That's right. There are two round bacteria, which makes it diplococci. Click on any of the bacteria and identify its shape. You can always look at the theory or image tabs if needed. Which shape is the bacteria you caught? That's right. This bacterium is corkscrew-shaped and so it's a spirochete. Click on any of the bacteria and identify its shape. You can always look at the theory or image tabs if needed. Which shape is the bacteria you caught? Correct. This is a bacillus bacterium since it's rod-shaped. You are a legend. Great work. I once read that some bacteria with certain shapes, such as Vibrio, are more commonly invasive than others. Do you want to know more about bacteria with the same shapes as those found in our sample? A bacillus is a rod-shaped bacterium, typically with one or more flagella attached. It's one of the most motile types of bacteria. Escherichia coli, commonly abbreviated E. coli, is a gram-negative bacillus-shaped bacteria. It's one of the most studied prokaryotic organisms and is frequently used in biotechnology and microbiology studies. Many strains are harmless and it's a common inhabitant of the lower intestine in humans where they play a natural role in normal gut function. However, certain strains are pathogenic, frequently infect humans through fecal-oral transmission causing serious food poisoning, septic shock, meningitis, and urinary tract infections. The gram-positive bacilli bacteria, Listeria monocytogenes, is usually the cause of listeriosis, a disease acquired by eating food contaminated with the bacteria. The main clinical manifestations are sepsis and meningitis. The disease can be fatal for pregnant women, newborns, and immune-deficient individuals. Do you want to know more about bacteria with the same shapes as those found in our sample? Diplococci are round bacteria that are formed from two joined coccus cells. You may have heard of the sexually transmitted genitourinary disease gonorrhea. The disease is caused by gram-negative diplococci bacteria called Neisseria gonorrhoeae. Another diplococci bacteria you might have heard of is Streptococcus pneumoniae. It's a gram-positive bacteria typically found in pairs as diplococci. In healthy individuals, it can colonize the respiratory tract asymptomatically, but it is also recognized as the main cause of community-acquired pneumonia and meningitis in children and the elderly, who often have a weaker immune system. Do you want to know more about bacteria with the same shapes as those found in our sample? Spirochete are motile gram-negative bacteria which resembles a telephone cord. They are unique as they have endocellular flagella which propel them around. They are characteristically found in liquid environments like mud, water, blood and lymph fluid. You might actually know some spirochete bacteria that causes diseases. For example, the disease syphilis is caused by the spirochete bacteria Treponema pallidum and is most commonly spread by sexual activity. Another disease caused by spirochete bacteria is Borrelia, or Lyme disease, which is caused by Borrelia burgdorferi. This disease is transmitted to humans via the bites of infected ticks. Do you want to know more about bacteria with the same shapes as those found in our sample? Vibrio bacteria are comma-shaped bacteria, typically with a flagellum at one end. You might have heard of the disease cholera which causes diarrhea and severe dehydration. Vibrio cholerae is gram-negative and colonizes the small intestine and releases enterotoxins. Many antibiotic medications are ineffective in treating cholera. Another Vibrio bacteria is Vibrio vulnificus which is gram-negative and motile. It's an extremely virulent bacterium that can cause acute vomiting, diarrhea, and abdominal pain and also severe sepsis. The infection is usually caused by eating undercooked or raw shellfish or through having open wounds in aquatic environments. Do you want to know more about bacteria with the same shapes as those found in our sample? Awesome! Bacteria, such strange little organisms. They come in so many shapes and sizes. Let's jump back to the lab. Well done. You have completed the simulation. Today we have learned how to look at bacteria with a microscope and how the shape of a bacteria affects the way it moves. The information you collected has been relayed back to Nicholas. Let me ask you one last question. Do you feel like you gained relevant knowledge during this simulation? Thank you for your feedback. Your score has been saved together with your 100% progress. Congratulations, you have completed the simulation. We hope you enjoyed it and you feel wiser now.